presented by Caltech.
It's my great pleasure to be here and really appreciate everybody to join the talk. I would like to walk you through my journey from the grad school, as Michael just mentioned, coming from a genome editing sort of world, and then moving towards single-cell genomics, and really trying to combine these technologies for understanding immunology and the immune system's regulation, particularly the context of tumor immunity. So really what inspired me to start tackling and really getting into the field of bioengineering or genome editing was because of the challenge when I kind of got into grad school and started to really do real biology research. I was both amazed and really also kind of daunted by the complexity of our biological system. So this is sort of the picture of insulin signaling pathway, very well known and well characterized. I remember when I was in high school, the textbook has maybe a third of the components that's known now, and this is probably not even an updated version of the signaling pathway as it stands now. But we can appreciate the complex regulation in the intracellular signaling space. And similarly, when we see cell as a single unit, we also have great complexity because even a single population of cell or a single cell type, as we define it now, consume a great variety of different states. This may not be super clear, but on the top is mouse T cell regulation, the differentiation pathway of different subtype of mouse T cells in the bottom of the human T cell. From this, I want to demonstrate that as we learn more about systems such as immune system, we appreciate the cells consume different states, even similar, identical cell types. And also there are differences between different species as well through evolutionary, changes. So the complexity of this biological system pose a really great challenge for us understanding their biology and how they're regulated in health and disease. So that's the first challenge. And the second challenge is even after we have sequencing data from genome-wide association studies, we have identified from, for example, 4 genetic mutation studies, key players and different cell states based on, for example, surface markers, we have a problem of how we can systematically validate, establish causal relationships between the signals that we observe versus actual phenotypic readout or basically manifestations in the biological system because there are so many different cell types and so many molecules involved. So these two challenges, I think, requires a scalable approach of bioengineering where we can really exhaustively, in a scalable fashion, understand the biological system using new tools. And that's really powerful because if we have the ability to systematically understand the expression pattern, for example, of different genes in different cell types at high resolution, and also perturb each individual elements in the similar fashion, we'll really start understanding the complex systems such as the nervous system, which is composed of different cell types, and also each individual cells are talking to each other through this complex network. As well as applications where genetic disease, sometimes really untreatable, such as sickle cell anemia, because of genetic changes, can be reversed and kind of engineered in gene therapy approaches that really help to improve human health. So with these two kind of challenges and the possible desirable technology in mind, I want to introduce today's my kind of journey into this technology and biological study in 2 parts. The first part, mostly my work from PhD phase in Feng Zhang's lab, working on developing multiplex and scalable genome perturbation technology in mammalian systems. And the second part, pushing further by combining a transcriptomic analysis with the genome editing to really understand the immune system's regulation during tumor progression. So from technology, kind of slowly getting down to biological insight. So the first part is genome editing technology. The complexity of the system really motivates us to think about how we can really engineer a technology that matches the scale of the biological system. We cannot always stay with a single target validation or single target engineering process if we want to understand the complex scenario as I have just showed you. So we want to perform genome engineering. So before getting into the actual study, I want to quickly go over the principles of genome editing. So this is a sort of an image showing the state-of-the-art genome editing process. Essentially, you 3D print a piece of nucleic acid and use a tweezer to kind of move things around. Well, that's not really the case. So this is not really what we're doing. The genome editing really takes advantage of both a designer DNA modification in the form of double-stranded break or single-stranded nick, and using endogenous DNA pathways such as non-homologous end joining or homologous directed repair, we can precisely knocking out a piece of DNA, generating random indels, or perform very accurate DNA replacement using recombination. This is really work pioneered by a lot of biologists studying DNA repair and DNA damage. So when I got into grad school around year 2010, we were really looking to take whatever we have at the moment instead of reinventing the wheel. So we first into zinc finger proteins, which is a great system for engineering the genome at the time, and pioneered by a lot of biologists and technologists. But the problem is zinc finger proteins are not very reliable. It's also commercially and even sometimes academically controlled by a few companies like Sangamo Bioscience and Sigma that is not quite accessible to us. So because of the limitation of zinc finger, we have to, instead of using whatever is available at then to engineer the genome, to develop our own system, which is the tail effector protein system. And among other people's study, the first part of my grad school really focused on how to harness this very intriguing bacterial DNA binding protein as a versatile genome editing tool. So because of time, I won't go into this part of study. But basically, this gives us a very simplistic system where by simply changing DNA's protein sequence, we can recognize all the 4 different bases to some extent of accuracy in mammalian cell. So we got really excited. And we think now we are ready to go scalable. We wanted to apply this to a lot of different systems and even think about we can use this to systematically map out using a tile library every single gene's function in a particular biological system, such as the survival of a particular cell type. However, we ran into the problem. So is this system really solving our problem? Is it convenient to build and very fast to deploy? Is the cost reasonable? Or is it really scalable? Because for every single sequence, we have to build a de novo build a new protein. So in a way, it's a really good system, but maybe not for everybody. Showing an example here from California. And so that's why we keep looking. We keep trying to find a better system that have true scalability. And that's the first part. I want to focus on the CRISPR-Cas9 system and briefly introduce my graduate school work. So as most of you probably know, CRISPR is the RNA-guided nucleic acid recognition system originally discovered in bacteria. And without going into detail, I just want to particularly mention that the CRISPR system really is work from an entire community involving many different scientists across the world to really figure out how the bacterial system works for us to really be able to engineer for mammalian application. And because of the pioneering work from all these biologists, we're able to work on how can we harness this particularly powerful bacterial system in mammalian cells. And we have to overcome a series of obstacles, which is kind of a recurring theme in bioengineering. First, bacterial system doesn't have any nucleus, compartmentalization. So the first barrier was targeting it to the nucleus. So we optimized the localization signal in different components of the CRISPR system, and we're able to really put it in the place where we want to target those proteins, namely the nucleus of the mammalian cell. And the CRISPR system is composed of RNA-guided proteins. So after the protein part, we also have to worry about the RNA part. In the bacteria, actually from Chamatier's work in 2011, we know that there are multiple forms of RNA expressed from the bacterial locus. Are these all functional? Or what is the minimal components? So because we want to, in engineering, for actual application, we want a minimum system that works well. So we mapped out what is really a functionally expressed and stable components of RNA in the mammalian cell system, shown here in the western block, in the northern block. And we realized that only one form of these sort of tracer RNA was really stable in mammalian cell, and then allowed us to combine the different parts of the system together to test out whether we can re-target or re-engineer the system to program it to target mammalian genome elements. In this case, a gene EMX1 in the human genome. So we test different combinations of actually 2 different Cas9 systems in our initial study, and we're able to map out the minimal system that works in mammalian cell, which is composed of a Cas9, a tracer RNA, and a CRISPR targeting RNA. The original CRISPR system also requires a RNase, which is not required in mammalian cell, as it turned out. So we have now the minimal working sort of CRISPR genome targeting sort of complex in mammalian cell. The power of this system really lies in the scalability. So with this system, not only can you simply reprogram RNA array to target any sequence of your choice, but also because of the natural multiplicity of the system, the CRISPR can be formatted in an array fashion and perform targeting of multiple different genomic locus at the same time. And we're also able to show not only recombination, but also precise deletion of multiple locations in the same time. So this system really allowed us to achieve what we were hoping to have in the beginning when we started looking at zinc finger, a scalable and multiplexable genome editing system in mammalian cells. And of course, with many people study, particularly actually similar in parallel, there were efforts from George's lab as well to show that we have different modality of the CRISPR system. We can simplify it even to a single RNA with the Cas9 to perform genome editing. And that really gave us the power of having an RNA-guided, highly programmable DNA elements, DNA targeting system in mammalian cell with a minimal functional components defined. And particularly critical, there is some diversity of the system, either at the design modality and also as I mentioned, there are 2 different Cas9 systems that actually we were able to show that works in mammalian cells. So there are a lot of sort of repertoire of the system for developing new molecular tools, the potential of that. And lastly, the multiplicity and scalability of the system because of the easiness of synthesizing nucleic acid. So these 4 components were really key for the later development in the field. And for really diving into some of the newer development, I want to show quickly some of the 3 more recent work I did towards the end of my graduate school on both applying CRISPR system for in vivo gene editing, which transitioned well into my current postdoc work, working with the immune system, and then also how we can also improve the system through structure-based engineering. And lastly, going back to the scalability that really motivated us in the beginning, how we can apply the system for a genetic screen. So going into the first part, we know the Cas9. I just mentioned that there's a lot of diversity. And the critical thing about diversity is because for different applications, we might want different type of characteristics for the system. And one system might not fit all the applications. For example, for one of the really cool applications I mentioned in the beginning, for somatic genome editing in vivo, we have to deliver the system into a body. So the barrier not only is we have the system that works, but also convenient for delivery. So one of the things that we wanted to do in the beginning to demonstrate this is to editing the mammalian genome with a AAV-based delivery system, which is one of the most promising gene therapy vehicle because of low immunogenicity, repertoire of delivery specificity, so different AAV serotypes can target the brain, liver, heart, muscle for different purposes. And also, not only has demonstrated clinical application, but for a variety of biological studies, this is ideal delivery vehicle for research. But the problem becomes the size limitation. AAV naturally can hold about 5 kb of cargo, but the minimal ASP-Cas9 system that works really well for us is over the delivery limit. But think about the solution. We actually go back to our first study. We were able to show there was one more Cas9 that works called ST-Cas9. This is actually in the supplement, so a lot of people probably doesn't even notice it. But this gives us the promise of thinking about whether we can export the diversity of the system for engineering a natural occurring smaller Cas9. animable for delivery. And here we take a serial approach where we first perform a metagenomic mining of different Cas9 systems and in vitro characterize the property of these systems to find the most promising candidate. And finally, combine them with the state-of-the-art delivery vehicle and animal model to demonstrate its efficacy for in vivo genome editing. And to quickly go over the full line of story, the natural Cas9 system have a lot of different varieties. And interesting, when we plot their size distribution, we have two clusters, actually, of Cas9. One bigger ones, including the classic SP Cas9, and a smaller cluster with the ST. The problem is the ST, which we already did demonstrate earlier, is not very efficient and reliable in terms of editing. So we went for some of the other Cas9 systems naturally occurring. So we tested a variety of different Cas9 systems. And particularly interesting is in vitro, we have a lot of systems working to cleave, but in vivo, in mammalian cell, we only have one. Actually, these two are the controls from SP and ST. Only one Cas9 system called SA from S. aureus Cas9 really worked robustly in mammalian cell. So with that Cas9 in hand, even though we're not that lucky, we only have one, we went ahead with testing it in vivo by packaging the system using a AAV delivery vehicle and targeting a therapeutic relevant gene called PCSK9. So this is a gene that's discovered through human genetics to be critical for regulating blood cholesterol level. And people have shown that if you block the activity of this gene in human patient, you can actually protect the patient from cardiovascular disease by lowering the blood cholesterol. So we want to test if we can achieve this with CRISPR in vivo editing. So we make the virus, and quickly, instead of relying on, for example, a transgenic mouse, which takes months to make, we can, within 2 to 3 weeks, perform an injection in live animal and perform genome editing in vivo in the tissue. So by tracking the animal at different time points, we're able to show that even within 7 days of injection, we're able to achieve very reliable and high efficiency genome editing with both guys that we tested on PCSK9. We were able to actually knock down the protein level. So the gray line is the control, which has roughly the same level over time, but at all the dosages we tested, even the lowest dosage, which is actually below the safety level of equivalency human dosage tested in clinics, we're able to achieve over 90% protein knockdown of the PCSK9 in the mouse serum, and a corresponding over 40% knockdown of the blood cholesterol level in the same mouse, which is actually as good as the best monoclonal antibody we're able to achieve in mouse model. So this really demonstrates us the systems works quite well. So efficacy proved. But is this kind of ready to go? We have a system now that we can deploy to any genome editing task? We don't think so, because as many of you probably know, the genome editing has suffered from a lot of doubts about their specificity. And previous study rely largely on sort of in silico prediction of sort of specificity of target sites. But for in vivo efficacy, we want to test unbiased genome-wide sort of understanding of the specificity in vivo. So we developed a new assay called BLAST, which adapted from an assay people use for map double-stranded break for sort of DSP hotspot previously. And we were able to adapt this system for Cas9-induced DSP detection. So the concept is very simple. It's kind of like a chipstick, where we basically have cells that are gently fixed to preserve their double-stranded break in their nucleus. And we like it on a biotinylated adapter that allow us to enrich for only the double-stranded break sites in the cell and perform massive parallel sequencing to map out systematically without any bias or selection what are the bona fide double-stranded break sites in the cell. And a small sort of vineyard is, when we first got the data, we were saying, that's great, let's just look at whatever signal we have, but we saw signal everywhere. That's because in natural cells, there are a lot of what we call DSP hotspot, particularly, for example, around centromeres, probably during cell division, as well in the telomeric region, similarly. So we have a lot of noise. Through a computational pipeline, we actually distinguished this background sort of double-stranded break from Cas9 induced double-stranded because of the pattern of the reads. You can see in natural double-stranded break hotspot, the distribution of the reads are fairly random across the region. Whereas because the capture method we use are very precise, we have very nice distribution of the reads at the junction site of the Cas9 cleavage. Also speaking for actually a good evidence of how the Cas9 cleaves the genomic DNA. So we were able to identify this Cas9 induced peak through this approach, and here shows the results for two different guides, where we're comparing actually on the top panel is the results from SA Cas9, and in the bottom panel, showing in the red color, is the results from SP Cas9. And we're specifically targeting the same locus shown here. So using the same type of guide sequence. And this chart, the way to read this chart is Manhattan plot, which is different chromosomes on the x-axis, meaning this is chromosome 1, 2, 3, so covering the entire genome. And on the vertical axis is the level of DSB signal we detected in the sequencing run. And you can see this is on-target site. We have a fair amount of really good consistent on-target modification for both Cas9, consistently between the two. Whereas for both locus, we saw, for example, cases where both Cas9 have off-target because we're targeting the same sequence here, which is expected. But we seem to observe much more off-target events with SP compared to SA, which is actually a very surprising but nice finding because this seems to indicate to us assay is a very accurate, potentially more specific Cas9 system from this unbiased assay. And further, we go in vivo to look at possible DSP formation in mouse models. And here, this is on-target, same assay with the two guide we used for PCSK9 in the actual mouse experiment. And we also perform deep sequencing of the top locus from the BLAST assay, and validated using basically kind of Illumina sequencing to see if this is bona fide, really no indel generated. And we see no indels actually in any of the sites that we were able to sequence, at least to the detection limit. So we were really confident that this assay, at least to some extent, captures the specificity of our assay Cas9 system in vivo. And corresponding to this test, when we perform histological studies, as well as tests of key sort of physiological enzymes, this is our AST level, and AST level, which is indicator of liver function. We also observe no differences between the control or the AAV-GFP-induced Cas9 or the normal sort of non-injected group. So speaking for the specificity of our assay Cas9 system. So even though more assays probably needed for more translational work, we think we have, through a very nice diversity exploration approach, identified new modalities where we can explore the natural sort of variety of different Cas9 systems to suit our specific need. And more importantly, this type of technology opens a lot of space for various applications. So since our publication, many people have used our system, most recently on a mastrophy disease model to show the rescue of this assay Cas9. And they were actually only able to show robust editing with assay because of the small size of the system allow more efficient delivery. And the second part is about further engineer the Cas9. So the first part is kind of natural diversity give us ability to engineer the system. And the second part is really about our sort of exploring the system's own property to artificially engineer the system to improve. And here we will rely on the structure information. Previously, we were able to solve the SP Cas9 structure. I was involved in the resolution of the SA Cas9 structure, where we basically were able to obtain the crystal structure of SA Cas9, but compared with, so this is on the left, SA, and on the right is the SP. We can very nicely see the differences between the Cas9 system, and this kind of pale blue part shows you how the assay were able to both have a miniature size, but also compare the two structures, showing that it removed a lot of the parts that are not contacting the nucleic acid or the guide, but preserved all the critical parts that are either interacting with the guide or effecting. So these two domains are the nucleus domain. So actually only removing the parts that are potentially not relevant for the genome editing function. And through the understanding of the structure, we're able to actually engineer new modalities very efficiently into the assay Cas9 system. In this case, looking at the structure, we can know what are the parts of the protein that are accessible. And we can attach basically different modality of either protein modules or RNA binding modules to this complex without affecting its genome editing sort of ability to enable a very efficient crystal-based engineering of transcription activator based on SA-Cas9. Unlike SP-Cas9, where a lot of times we have to do optimizations through series of constructs, in this case, we only did very targeted trial based on the crystal structure, and we're able to achieve very good results even on the first go. And shown here is our functional test where we have a GFP showing the expression of the construct and the MKit reporter showing the transcription activation level. And with the non-targeting guide, we have nothing activated, very low background. And with the targeting guide, with the assay activator system, we're able to achieve very high level of gene activation, both for reporter construct shown on the left and also on the right, a very potent activation compared to the SP system in mammalian cell for endogenous gene. So without any optimization, we're able to achieve very high level of sort of activation even in the first construct we build. So speaking for the power of getting the molecular detail. And similarly, for control the activity, we also have been able to split the SA-Cas9 based on the crystal structure, only designed 3 different constructs shown here. And actually, one of them is specifically designed to serve as a control. You can see here, we designed 2 constructs breaking on the loop region, which we believe will make the Cas9 splittable, and one particular control in the middle of the protein that we thought will disrupt the connection. And indeed, the 2 kind of targeted, the 2 kind of structure-based design really worked well, and the kind of this intentional control had no activity when you reconstitute the parts in mammalian cell. So we now can achieve a very nice sort of inducible system using the split Cas9, where we fuse inducible domains to the Cas9, and we're able to achieve inducible gene expression using chemical inducers. So this also has very minimal sort of test or evolution involved because we have very nice available crystal structure information. So this part really shows the ability of using the Cas9's own endogenous kind of intrinsic property to guide our engineering efforts. And following this 2 story of improving the system, let's talk about sort of the scalability of the system that really motivated us in the first place. And I want to go quickly this part because I think a lot of you are quite familiar with the story. The benefit, even before we actually make it work, we know that the nucleic acid-based system benefits from the massive parallel synthesis ability of nucleic acid sequences that allows you to build a library of CRISPR targeting guide to package into delivery vehicle for massive parallel screening, meaning each individual cell will serve as a small experiment to allow us to read out biological phenotypes if we have the proper assay. And demonstrating, this is mostly work that I was involved from our colleagues in the lab, Neville and Fear, where they performed this screening on a melanoma model that we know melanoma grows very nicely, without any sort of drug treatment. But when you have this targeted drug, BRAF inhibitor treating BRAF mutated version of the melanoma, we can really nicely inhibit the growth of the tumor, but they can also develop resistance to the drug in the process. So in order to pin down the resistance genes, we perform kind of a genome-wide screening, comparing the enrichment of the guide with the selective pressure versus without the selective pressure. And this approach really identified through enrichment of sgRNA what are the candidate genes that are critical for regulating this drug resistance of these cancer cell lines. And more critically is with the scalability of the system, we can also imagine exploring the diversity of the functionality that we have been engineered, as I showed you earlier, to expand it to more applications such as transcription activating screen, also demonstrated earlier in the lab. So for this part, to recap what we have developed is through enabling a very scalable system with a lot of applications in mind, we're able to serially engineer the system exploring different modalities and demonstrating both the versatility of the system as well as the scalability of the system. So with that, I would like to go to the next part, really focusing on my postdoc work in Avi's lab on tumor immunity, where we applied both the genetic perturbation as a validation module, but go into the biology of the system by characterizing the transcriptional signatures of tumor immunity. So going back to the challenge of So why we're going with this transcriptome followed by sort of genome perturbation approach is because, again, the complexity of the system. The cells have different state, and we don't necessarily know the key molecules that define these states, even though we have a few marker genes that lay out by previous biological studies. So for really comprehensively map out the regulations of tumor immunity, we want to take a whole systems transcriptomic approach. And as you know, cancer immunology is probably the best place to apply this type of approach because in the scenario, in the cycle of a tumor progression, there are various phases where the tumor is initiating, and then immune system will start to act and locate the tumor, killing it, but then tumor were also able to counteract through regulating our tumor immunity and prevent the immune system from destroying the tumor and allow the tumor to grow in the body. So in this part, I would like to focus on two very recent stories we've sort of completed in the lab. The first one is analysis focusing on tumor-infiltrating T cells and how their regulation is critical for killing versus dysfunction of the killing phenotype or dysfunctional phenotype of these TILs in a melanoma model. And second part is going into the single-cell analysis to understand tumor immune set heterogeneity as well as its interaction. So going to the first part, so this is a classic problem where we know a lot of T cells can recognize tumor, but they got regulated by the tumor microenvironment and become what we call dysfunctional. So we used a model in collaboration with Vijay Kruchu's lab at Harvard Medical School, where they have the model called transplantation melanoma model, where you can basically have a melanoma cell line, a mouse tumor melanoma cell line that you can inject into a healthy mouse so that you can basically give the mouse a growing tumor cell. And because it's from the same genetic background, the tumor will be recognized by the immune system and start to mount an immune response to the tumor. And as the T cell infiltrates the tumor will able to actually counteract this process by effecting a dysfunctional T cell phenotype. And previous study in Vijay's lab defined functional molecules defining these different phenotypic changes in the T cell with what we call double negative T cells, single positive T cells, or double positive T cell defined by two key markers, TIM3 and PD-1. These are what we call co-inhibitor molecules. Basically, this molecule function to inhibit T cell function once they're activated. And so this molecule expression on the surface of T cell define different functional state of the cell as previously measured in the Vijay's lab by secretion of key effector molecules. For example, in the double positive T cell, highly expressed TIM3 and PD-1, there's very minimal secretion of effector molecules, but actually secretion of anti-inflammatory molecules such as IL-10. But in the double negative T cells, they have very strong secretion of tumor-killing cytokines such as interferon gamma and TNF alpha. So by isolating these T cells, guided by this existing marker, what we want to achieve is to define what key regulators are differentiating between these different functional states. So what we did is we take this population through classic marker-guided sorting so that we can separate them and perform a whole transcriptome analysis using RNA sequencing. And then we look at the differentially expressed gene in this different population shown here. So we have actually three different populations and two functional controls. The three populations as defined earlier, and also two functional controls one representing naive T cell, which are not activated, and what we call effector memory T cell, which are activated T cells defined by classic functional marker. And you can see, through looking at differentially expressed genes, the double negative, or the more functional T cell, really resembles actually the effector memory T cell expression profile. Whereas the double positive T cell, which is dysfunctional, will assume a different transcription program, very distinctive from other types. And from that, we actually find out there's a very top candidate that consistently show up as differential regulated between the different cells called MT gene family. So this family of genes are zinc binding family of protein that the function is actually not known. And previous data have some initial idea that these genes are critical for the function of T cell. So taking a conventional approach, because we have a transgenic line available, what we did is we performed a transgenic mouse study where we take a knockout mouse cell line, knocking out the empty genes by traditional transgenic technique, and compare the tumor growth in both wild-type background and empty knockout background. And we see, indeed, this is confirmation, as expected, that in the empty background, there is improved T cell function. I should mention that the empty genes enriched in this is highly expressed and enriched in this cluster, which means that it is highly expressed in the dysfunctional T cell. So we expect when you knock down this gene expression, you will have rescue of that dysfunctional phenotype, leading to a better tumor killing. So this vertical axis is showing you the tumor growth, and we have less tumor growth when we have the empty knockout. So this is a classic approach. But how about all these other genes that we discovered? How do we really systematically map out the entire program dictating the T cell function? So now we are going to more scalable approach. So we first want to dissect out what are the different genes that are really defining the functional state. Because we have this classic empty knockout example, we did a comparative study shown here where we want to further dissect the different functional state of the cells. As we can separate, we have previously characterized the three population from the wild-type mouse. We also have the ability to perform sim analysis by isolating cells from the empty knockout background, which means these cells, based on, or at least partially, are being rescued from the exhaustion state or the dysfunctional state. So they are removed from the dysfunctional regulation to some extent. And we compare using RNA sequencing the different transcription profile of these distinctive populations. We're able to map them using a principal component analysis to a two-dimensional space and look at what are the defining gene families that are explaining the variabilities of these genes, these different populations' expression signature. So on the left is a mapping of all different examples. But most strikingly, if you separate the wild-type example and the empty knockout example, we can see that the two top PCs, the PC1 here and the PC2 here, can explain distinctive variability in the examples. First, the PC1. The PC1 actually nicely separate the different individual groups colored by, shown here in different color, in both wild-type examples and in empty knockout examples. As you can see here, the red is double positive, the more dysfunctional cell, and the black is the double negative, the more functional cell. So in both wild-type and empty knockout, the first PC were able to separate the different examples, explain the variability. So this family of genes, heavily loaded on the PC1, are explaining the variability in both samples. But the most interesting one would be the PC2, because the PC2 only captures the differences in the wild-type sample. Meaning that if you remove the regulation from the empty family, you remove these genes' effects over the T cell. So what we think that we want to test as a hypothesis is how about, this is a computational hypothesis. I want to emphasize that we want to test if it's true that the PC1 from the empty knockout group, which basically is consistently explained in both wild-type and empty knockout, explain an activation score. Because as I mentioned, both these cells are activation, both these PC1 genes can differentiate between activated cells versus dysfunctional cells. Whereas the PC2 can really explain the dysfunctional state. And we basically mapped expression level of all different genes into these two axes by their expression level, correlating their expression level with the two different axes defined by the activation score and dysfunctional score in terms of the loadings of the different genes observed in PC1 in empty knockout and PC2 in wild type. And in this axis, because the rationale is being, if you have genes expression more correlate with the PC1, which with separate activation state of the cell, you probably are capturing activation genes on the one axis, and think about the number 2 PC, which really only separates cells in the wild type. And remember, the empty removes the dysfunctional state, presumably at least to some extent. So we think the PC2, which capture wild type sample, will represent the dysfunctional differences between the cell, which was removed in wild type. So if you correlate these scores to the PC2, you can probably define this as correlation with dysfunction. So if you map out these 4 different quadrants, you can see here on the top left is dysfunctional only genes. On the top right is activation and dysfunction. And on the bottom right is the activation only genes. So by making this correlational study, we want to see if this is really true. And we particularly want to validate the results from the top left axis, because these are genes particularly involved in only dysfunctional regulation. And so this red color are all the TFs that we mapped in this quadrant. And we want to see if we can validate this, focusing on one of the genes shown here, GATA3. We focus on this gene because this gene is now known to play any role in CD8 T cells. It's actually known to be critical for T cell differentiation. So actually, when we obtain some knockout mice and use the GATA3 knockout mice to perform the similar experiment we did with MT, we don't see any particular difference. However, we want to see if this is because GATA3 has differential role in different phases of T cell development. So what we want to do is a particular precise timing of the perturbation using CRISPR system, where we have a particular mouse called PML mouse that recognizes the melanoma cell using our model, and we can take basically naive T cell from the mouse and use CRISPR to knock out the GATA3 gene after we activate these cells, and then we can refuse or basically inject back this T cell in a turin-bearing mouse. Because the turin-bearing mouse is wild type, the tumor will be able to counteract the T cell infiltration and the killing effect and growing. But what about if we give them these perturbed T cell, and whether we can induce a phenotypic change? And indeed, even though we don't see an effect in transgenic traditional knockout mice, we're able to not only significantly knock out the gene using a CRISPR targeting the GATA3 gene, but also observe a very significant sort of phenotypic shift in tumor growth in vivo when we transfer these GATA3 cells, which are not observed in the traditional knockout. And we think this is really because of the timing of this knockout in precisely giving this perturbation at a regulatory process. And we're now following up with more in-depth analysis in this experiment to understand the molecular regulation that's involved, that it can explain basically why GATA3 is critical for defining T cell dysfunction. And to wrap up this section, I want to take this as an example, but demonstrating to you that because of the scalability of the CRISPR system, we now have a very nice workflow that allow us to not only perform this single target validation, but think about scalable sort of applications in T cell population using a pooled library screening approach, which we have demonstrated previously in the lab for dendritic cells. To summarize this part, we really take a guided approach by classic functional marker to perform systematic analysis on T cell gene expression to compare different T cell state to identify potential candidate genes that regulate their function, and using a scalable approach based on CRISPR-Cas9 system to precisely perturb these cells and demonstrate biological regulation in vivo. And finally, I want to quickly walk through a more recent work, which is still ongoing in applying single-cell analysis on tumor immunity, particularly on dissecting out their interactions between tumor cells and immune cells. So in this case, we're collaborating with Tyler's lab. This is a very new collaboration I initiated when I came to Aviv's lab, where the Tyler lab has been focusing on a genetic engineering model. I want to quickly walk through this model because it has very distinctive benefit from the model that I just showed you based on transplantation. Their model is based on genetic engineer tumor-initiating mutations, KRAS, P53, into a mouse model. And the whole system inducible by Cree, and also labeled with a tomato, Cree-dependent tomato locus so that when you inject a Cre virus encoding both the Cre recombinase and also an artificial antigen, shown here is the luciferase over SIY artificial antigen cassette, you will be able to both induce at defined time points the oncogenic mutation in the mouse, and also label these cells with tomato fluorescence so that you can isolate the cells out, and also give them a specific strong antigen to trigger a strong immune response in the adaptive immune system. So this allows us to dissect both tumor initiation and progression, which is now actually quite possible with the transplantation model. So this captures the whole life cycle of the tumor. And most critically, previous biological work on the phenotypic characterization of this mouse showed us the immunoediting of this tumor in the process of progression, meaning that the tumor will initially be sort of infiltrated by the T cell, stop their growth, but then they can escape the immunoediting without losing their antigen by regulating the immune response and really overcome the immune surveillance and suppression to keep growing and into adenocarcinoma, which is a great model for non-small cell lung cancer, which is a very prevalent cancer type in human patients as well. So what we did is we want to go into this complex system to really dissect the regulation and heterogeneity by using a time point longitudinal study, where we can basically track this tumor growth at key time points, 8, 12, and 20 weeks. Basically, the initiation phase, the peak immune response time point, and also the exhaustion time point at 20 weeks to understand the whole process. Additionally, we focused on the tumor and T cell population in this model because we have a very strong tumor antigen. So we isolated the tumor and T cell population from the lymph node and also the lung tissue as a comparison between relevant tissue and tumor-bearing T cells, and truly try to understand the heterogeneity of the regulation of the cells using single-cell genomics, because we believe there could be significant heterogeneity that are missing if we just perform the same type of bulk analysis using the traditional method, and also because we don't have a well-defined marker for this model. So I want to show you some initial set of data quickly, where we focused on T regulatory T cell, Tregs, particularly from the lung and lymph node. And we also have a CD8 T cell population as a pilot. Shown here is that our protocol of, this is about 200-ish single cell total from each population. And we were able to show that the single cell data correlated really well with our population control, speaking for that our process really recapitulated the majority of the transcription signature of the cells. And nicely shown here is that we can also using single cell to capture the identity of different populations faithfully by using simple PCE analysis and project all the top PCs to a TSNE space. And you can see here, these are different. So the red is the Treg example from our lung, and the green is Treg examples from our, the red is the CD8, excuse me. And the green is the Treg from the lung, and the blue is the Treg from the lymph node. And you can see we separate this population very well in the TSNE projection space. And also, if you look at marker gene expression such as CD8 and phospho-P3, which is a Treg cell marker, they really recapture the cell population very faithfully. And you can see the blue is a highly expressed gene. So, so it's really recapitulated this marker gene expression. And particularly interesting is we start to initially looking into the differential expressive genes, similar to what we did, but as a single cell resolution. So here is a cluster using the data from only the Treg between the lung, labeled here with the kind of the purple color or the magenta color, and also the blue color labels the lymph node Treg. And you can see very nicely that if we just keep using the highly differential expressive genes to color these cells, they fall nicely into basically lymph node versus lung. And most intriguingly, if you look at the expression of these top differential expressed genes, these include some of the really interesting markers of T cell regulation, both in co-distributed molecules such as CTLA4, PD1, and also very nicely correlated to my previous story, which is quite unexpected, GATA3, which has never been shown to have any sort of critical function implication in phospho-P3 T cell in a real biological context, at least to our knowledge. and highly differentially expressed only in the lung, sort of highly expressed in the lung part of the compartment that's directly interacting with the tumor cells in our model. And this is actually from the 20-week time point, which is already having exhaustion. And as we know, Tregs really particularly suppressing immune response in T-cell regulation. So we think this could identify subpopulations of Treg cells that are critically sort of defining a program of immune suppression. And you can see we really can pin down some of the cells that are really heterogeneous in terms of expression signature, even though at broad level they will look probably similar if we perform a population analysis. So in summary, in this part, we have an initial set of single-cell data that both show the validity of approach in identifying the different population, as well as really start to dive into the differentiating signature and heterogeneity of lung versus lymph node Treg cells in their regulation in immune suppression. The next step that we're currently ongoing is really analyzing the single-cell analysis data from both the tumor cells as well as the T cells, and also compare data from different time points to really dissect the regulation in the dynamics of tumor growth. And finally, we also want to apply the same type of validation sort of concept that we implemented previously in a population study. And lastly, I probably have some construct already made for the Treg story at least. So to summarize this part, I hope to present a sort of further extension from the technology development where we systematically used a sort of understanding the complete scenario of tumor immunity from a both systematic approach using transcriptional analysis as well as a genome perturbation using tools that are scalable, developed from the first part of my graduate school work. And in the end, I want to walk through some of the future directions I want to pursue after this. I think coming out from the technology background, I wanted to really dive further into enabling scalable technology for understanding complex biological systems. And this goes back to the motivation that really initiated my whole research effort from graduate school extending to my postdoc training. Particularly the three different parts that I want to build, focusing on both having a better sort of scalable and real-time ability to both profiling and recording gene expression at single-cell resolution, which I'll explain in a moment in more detail, and also take advantage of the scalable tool that we've developed based on CRISPR system to both perturb in a very high throughput fashion the genetic regulation and epigenetic regulation that defines the cellular state and signal. And finally, by integrating the knowledge from cellular signaling and single-cell resolution understanding of the biological system to build synthetic sort of signaling, sensing, and transduction modules to allow us to engineer particularly immune cell identity for tracking and perturbing the immune response in either healthy state for understanding the immune system regulation or in disease state for potential therapeutic application. So in general, I hope to really take an iterative approach between technology development, applying them to complex biological systems, but also take advantage of very nice information we learned from the biological system to guide or design of a technology implementation. Specifically, quickly, I want to walk you through some of the specifics of each individual sort of broad aims for developing really a profiling recording technology. We can think about how we can apply some of these very multiple functional and multiplexing tools. For example, based on the inducible CRISPR system, where if we have an inducible CRISPR system that responds to a trigger in terms of a gene expression, we can record the expression of the gene in the presence of an inducible trigger so that we can define when we can execute the recording and genetically encode the expression level of the signal into a CRISPR-mediated genetic change in the genome. So that's what we can call a genetically encoded gene expression information that we can actually read out after the completion of a biological process. That gives you the information and ability to go beyond a transcriptional analysis based on traditional RNA sequencing. Because that, in the case of what I have been doing in my postdoc, we have to take samples at individual time point. And you lose the ability to track the cells. And we can have confounding factors because we're not tracking the same system using the same identical sort of cell population. And secondly, building on the information from the understanding of the system, we can think about how we can also complement a targeted approach of recording or profiling with a non-targeted, unbiased approach using high throughput perturbation. As demonstrated earlier, if we have libraries of sort of elements that targeting systematically different interesting regulatory sort of genes or sort of elements in the genome, we can systematically map out in a defined in vitro in vivo system the regulatory networks. And then we can, using that information, to combine with single-cell profiling and guide or targeted recording efforts to really have this iterative sort of synergistic process for understanding precisely what's defined a biological state and signal, in particularly in the immune sensing response, which I want to focus as a biological sort of core of my future research program. And finally, not only can we bring existing knowledge, but also integrating potential knowledge from the first two aims, we can finally integrating existing knowledge of using a well-defined system, or if we can define a currently uncharacterized system using aim 1 and 2, we can imagine integrating a synthetic biology concept to build artificial circuit in what we can call a cell-based device that really rewires the signal from a natural cue and produce targeted, desirable response, either for outputting biological critical insight of the system or for applications in therapeutic intervention or diagnostic intervention in a biological setting by taking the signal, rewiring it using systems such as CRISPR to target a different set of response that we can hardwire into the cell in either in vitro or in vivo cases. So that really take advantage of the information, first two aims, and bring it to action. And hopefully with these three aims developing in parallel, we can really going back to the original drive of all my research efforts to study and understand complex system identity and regulation, as well as potentially apply these concepts and tools for applications such as gene therapy in addressing some of the most devastating human disease. And finally, I want to show you how much we have been achieved. Here, this is a chart of how many kind of CRISPR algorithms. I'm most amazed by how fast people have been really adapting it. And particularly in the context of Los Angeles, it can be sometimes too fast to give you some undesirable or cautious result. This is the problem sometimes of scalability, I guess, of a technology. But in the end, I really want to say thank you to all my advisors throughout these years. Without them and without some of the funding agencies that really supported my PhD work and from HHMI and also my postdoc work from CRI, I was really fortunate to perform all this work under the guidance of first initially with Feng and also our time together with George earlier, and particularly currently with Aviv, and in collaborating efforts with people in Tyler's lab and also in Kuchu lab. Thank you so much for having me here, and I'm happy to take questions.